So here's a quick overview of operons. I know a lot of people were struggling, so hopefully this helps. We can think of this as prokaryotic gene expression. So when we're looking at operons, we have to first understand the central dogma of biology, that DNA is transcribed into mRNA, which is translated into proteins. A lot of time those proteins are going to be enzymes, which means that we've got some kind of reactant yielding some type of product. In the case of gene regulation, typically if you have enough of a product, then that product is then going to be able to prevent transcription, and in that way this is a negative feedback loop, because when you have too much product, then you can do something to bring that level back down. On the reverse side, if you have a reactant that needs to be broken down, that is going to promote transcription, because then you're going to want the enzyme that's capable of breaking that reactant down. Okay, so we can go ahead and start by looking at the trip operon, or the tryptophan TRP operon. Same thing as before, we've got a protein, which in this case is an enzyme, catalyzing the reaction from reactant to product, and this is going to be a type of negative control. In this case, we've got precursor reactants that are then going to be used to make tryptophan, and tryptophan is an amino acid that is important for being able to make proteins. So if the cell does not have tryptophan, it needs to be able to make it. If the cell does have tryptophan, it does not need to waste its time. So we're going to fill out a little bit of a table here, and hopefully this starts to make it easier. We're going to look at the presence and absence, or plus and minus tryptophan, but I think it's going to be a little bit easier to look at it from ultimately what's going to be most beneficial for the cell. Not necessarily what the cell wants, but what is evolutionarily going to lead to a better survival rate. So if you have tryptophan already, are you going to want to have transcription happen to make the enzymes that make more tryptophan? And that's not going to make a lot of sense. So we don't want transcription. We want the gene to be off. And in this case, we're going to be looking at a repressor. Repressors are basically what pushes an off switch. So now we're making the gene turn off, meaning that we need an active repressor. Okay, so if you can think of all of that backwards, then you're in good shape, which means in terms of tryptophan, tryptophan has to somehow be involved in activating the repressor, which means that it is going to be a co-repressor. Tryptophan plus the repressor together is going to make an active repressor. On the flip side, if we don't have tryptophan, then we will want transcription to happen, because if we don't have tryptophan, then we can't make protein. So somehow we need to be able to make the tryptophan, and therefore we do want the gene to happen, right? So we want the gene to be on, we want the repressor to be inactive, and therefore no co-repressor, no active repressor, the end. Pause if you need to, take a second to take it all in, and I will move on now to the lac operon. In this case, we're looking at the breakdown of lactose into glucose. Lactose is not usable by itself. It must be broken down into glucose to be able to get energy out of it. So lactose, a disaccharide, glucose, a monosaccharide that can be used, and that reaction is catalyzed by beta-galactosidase, or this little guy that's right in right here. So when you have lactose present, then you will want transcription to occur so that you can break it down. We'll do the same thing, take a quick look at the table with and without lactose present. Now, an important note here, it's not actually going to be the lactose that is operating on the operon, but instead is going to be something called allolactose. Anytime you have lactose, you're also going to have a quantity of allolactose, so the presence of one indicates the presence of the other, and it's actually going to be allolactose that's playing a role here. In either case, if you have lactose, then you want to be able to break it down, which means you do want beta-galactosidase, which means you do want transcription, and you want the gene to be on so that you can make it. In terms of a repressor, remember, repressors repress. They stop transcription from happening. So if you have lactose and you do want transcription and you want the gene to be on, then you want to make sure that your repressor is inactive. So in the trip operon, we were looking at tryptophan as a co-repressor, tryptophan helping the repressor function. In this case, we want to do the opposite. Lactose needs to somehow inactivate the repressor, and that's going to make it what is known as an inducer. An inducer because it induces or causes transcription to occur. So with lactose, we do have transcription. Without lactose, there's no reason to make beta-galactosidase to break it down because you don't even have it in the first place. So we'll want the gene to be off, the repressor to be on. Now here's a little ditty. Lactose is kind of like a plan B. If you don't have enough energy in the cell, then it's really important that you can break down lactose. But what if you have a cell that has plenty of glucose, right? You've got lactose, but you've also got glucose. Plan A is going to be to use that glucose for energy. So the lac operon is under control of two different molecules in two different ways. Allolactose is going to act in negative gene regulation to downregulate whether or not transcription occurs, whereas glucose is going to play a role in the positive gene regulation. So again, we'll look at with and without glucose and look at the end result first. If you are a cell and you have glucose, it is evolutionarily beneficial for you to not transcribe beta-galactosidase. You have enough glucose. No need to try to make more, even if there's lactose present. So, no transcription, meaning we want the gene off. On the flip side of that, if you don't have glucose, you do want the gene on. So no glucose, then you need to go to plan B, start making beta-galactosidase so that your lactose can get digested. That means we need the gene to be on, and so we want to have an activator. An activator is just something that is going to bind to the promoter and allow for transcription to happen better. And that's the opposite of what we've been talking about. When we're thinking about a repressor, repressors block transcription from happening when they're active. In this case, activators are going to promote it, and so for that reason, it's a positive gene regulation example. Okay, so we need to activate CAP. CAP is the example of our activator in this case. And the way that we're going to do that is as follows. Okay, if you don't have a lot of glucose, low glucose concentration, that means you're not going to have a lot of ATP. Instead of ATP, our full battery, you're going to have a whole bunch of cyclic AMP, cyclic adenosine monophosphate, which is just essentially a really, really empty battery. What this is saying is that I don't have glucose and my cell is really lacking on energy, which means somehow I need to start making glucose so that I can get energy. So I need to be able to use my lactose to make the glucose using the enzyme beta-galactosidase. Okay, makes sense? So high levels of CAMP means let's get this show on the road. Cyclic AMP is going to bind to CAP, and when they're together, when CAMP is with CAP, that is what activates the activator, turning the gene on and allowing transcription to occur so that beta-galactosidase can be made, so that all the lactose that is present in the cell can be broken into glucose and you can make ATP, and then ATP levels rise and all is good with the world. Now, if you already have glucose, no need to go through all that rigmarole, because if you've got high glucose, then you already have high ATP, meaning you're going to have a low amount of cyclic AMP and your CAP is going to be inactive. Okay, I will post these PowerPoints just so that you actually have the completed tables. I also have posted the graphic organizer from Tuesday, so email me if you've got questions, and hopefully this made a little bit of sense for you. All right, bye.